as long as you know what happens. Okay, getting started, replication begins at the particular sites of called origin of replication. This is important. From here on is important. Up to this, I was just reviewing a little bit, but from here on, let's go ahead and today's material starts. Usually we review at the beginning, you give me the answer to the homework questions, but I just thought I'd review today. Okay, so what happens getting started, it means what? You have a DNA, you have one DNA that is going to multiply to 2, right? That's what I'm talking about. How does that happen? You should know that. Okay, so what happens, you have origin of replication. Replication means duplication. If you don't know what replication means duplication, where the DNA strands are separated, opening up a replication bubble. You saw it in the video, and then you will see it now, and I will talk about it. I'll show you a picture what a bubble means. Just hang on to that word, a bubble. Okay, and eukaryotic chromosomes may have hundreds of, even thousands of origin of replication. It means they have thousands and hundreds of bubbles. Replication proceeds in both directions from each origin until the entire molecule is copied, right here. If you forget about this one for a second, let's talk about plasmid. Plasmid, it means circular DNA. Plasmid means circular DNA. The latter, a circular DNA of the bacteria, and some protista are like that. Some bacteria, some protista DNA is round as a circle, and we call them plasmid. The DNA in eukaryotic cells in us is straight. It's straight. It's not circular. The DNA in eukaryotic cells, us, again I said some protista are eukaryote as well, but in the rest of eukaryotic domain, it is straight. Okay, so that's what DNA of, and then of course, now we are talking about the bubble. This is the bubble. This is the bubble, the origin of replication, and this is the bubble I was talking about. In a circular DNA, in a plasmid. In a straight DNA, you have several bubbles. As the last PowerPoint, as the last picture indicated, how many of them? Hundreds and even thousands of origin of replication. Hundreds or thousands of bubbles and origin of replication occurs. Am I making some sense, Tron? So what happens, the DNA is multiplying on both directions. The DNA is multiplying on both directions. It's not going on one direction, stop, and then another direction. No, going on both directions simultaneously. Maybe that's the right word. Okay, so the DNA is multiplying on both directions simultaneously. And there are some, of course, micrograph pictures in here. What is that? Origin of replication of eukaryotic, origin of replication, eukaryotic chromosomes, parent chromosomes, template, and then, of course, in this diagram, we refer to this one. You have to be familiar with it. This is the parent DNA, right? You remember this? Parent DNA, and then based on that parent DNA, this one is, they call it template. During exam, you ask me, Amir, what is template? I say, I don't know, I forgot. Because, why? Because I already talked about it. Am I making sense? So this one is the template also on this one. So you have a template and a new DNA. You have template and new DNA. New DNA is being formed. Okay, a little bit more details. Are you guys ready? I said, how many of you got my email? I don't want to expect you to check your emails 100 times a day. but a few of you got my email, right? And I had list of questions for you. If you don't have them, those of you who did not, but definitely by Wednesday, I'd like to have those homework questions back. If you have it today, fine. If you do not have it today, then hopefully by Wednesday. Until Wednesday, I try to send you another new set of questions. Okay.
You said plasmid is circular, and for humans it's straight. So
Our DNA,
our
yes,
DNA is straight. Would that be called anything special?
No, I do not have a term for it.
Oh.
Yeah, I do not have a term. Straight DNA.
Oh.
Okay, eukaryotic DNA. Usually referred as eukaryotic DNA.
Okay.
If sometime later on in your life you come across it. Okay, then okay, let's go. And then in that email, you guys, do you remember I sent you a set of terminology? You remember that? You need them today. I mean, not for the quiz. I didn't ask any quiz question. Your quiz question is from previous material. But today, I'm hoping you would you would know what we are talking about and so on and so forth. Okay, let's see what this video says.
Replication of a DNA molecule begins with a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of the eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
Okay? Same thing I said. Okay, at the end of each replication bubble is a replication fork Y shape region where new DNA strands are elongating. Helicase, I'm going through that list now. Helicase is the name of ACE at the end of the word. Is that right? It means what? It's what?
Enzymes.
Enzymes. Are enzymes that untwist the double helix at the replication fork. So DNA is like this. The DNA, before it duplicates, it has to become straight. Right? DNA is like this. You all know that. So DNA, before it becomes duplicated, they have to untwist. Who does this untwisting? Your helicase. And that was in your list, I hope. Is that right? And do you have that list with you? It's a PowerPoint set. Single strand binding proteins regions bind to stabilize single. These are protein molecules. These are protein molecules. When the DNA split, they attach to DNA that they do not. The DNA molecules do not come together. So DNA have to separate. When they separate, if you don't attach these protein molecules together to them, to one of the strands or both of the strands, the protein molecules, the DNA come together. Okay. And then the last one is missing, topoisomerase. It is, they will unwind, they correct and overwinding. They fix the overwinding ahead of replication fork by breaking, swelling, and replication. The best thing, I don't know if you guys have ever done this in your life or not. If you have a two rope, maybe we go to YouTube and I'll show you this. If you have two ropes and they're wind together, you want to separate them, right? Have you ever done that or not? I don't know. If you want to separate them, somewhere along, they will separate, but somewhere along the line, it jams. You know what I'm talking about? Huh? You're separating them. Helicase is separating them, and the binding protein is attaching, but somewhere along the line ahead of it, it will jam because the twisting. Okay, but this enzyme, topoisomerase, will go ahead and unwind those. Am I making some sense? So, and then as a result, helicase will come and then unwind those. These. Do you guys want to see the topoisomerase from YouTube? Should I go ahead and go to YouTube? I hope. Or no?
No.
No, good, thank you. All I need is one verdict. I don't need everybody approve it. That will take a whole year. Okay, right here, here's the topoisomerase. Is unwinding the DNA ahead of time, and here is helicase, okay, unzipping. DNA is like a zipper also. Imagine a zipper that is twisted, unzipping DNA, and of course you have, I will talk about primase here in a minute. Here is your binding protein molecules. These binding proteins attach to DNA, so DNA do not come together. I want to make this some sense a little bit. And then of course primase is the enzyme that attaches, makes RNA. This is RNA. The RNA is called the primer. Attaches and makes RNA to DNA. Okay. You might ask, Amir, why RNA? You all know the difference between RNA and DNA. We talked about that. RNA has uracil, RNA is single-stranded, DNA is double-stranded. A few other differences. RNA is ribose nucleic acid, DNA is deoxyribose nucleic acid. You might ask, Amir, why does it start with RNA at the origin? The reason is because the replication of DNA must go from 3' to 5. It starts at 3'. Okay, so only RNA can allow this DNA to have a 3' end, 5', and then 3', and then eventually, usually this U is between 5 to 10 RNA. After 5 to 10 RNA, then DNA nucleotides will come in place. The reason for that, again, you have a 3' end. Always DNA replication starts from 3' end. That's always starts from 3' end. Okay. Then if you have 3' end here, you must start with 5'. So replication, duplication of DNA starts from 5' to 3' end, because you have 3' there. I want to make this. Remember, we said DNA must be anti-parallel. It must be anti-parallel. If it is 3' there, then it must be 5' here. I started the lecture today with that. They are anti-parallel. 3' here, 5' here. And only RNA allows that for DNA duplication. Let's move on. And then I'm guessing if we move on and you see this more, it will make a little bit more sense. If you studied this before you come to the class today, you're getting every word I'm saying. Every word I'm saying, you're getting. If not, then you have to study tonight or I don't know, sometime. DNA polymerase cannot initiate. DNA polymerase is an enzyme that actually makes DNA. All of those enzymes I gave you so far, topoisomerase, helicase, primase, none of them make DNA. Those enzymes are initiating a replication of DNA. But DNA polymerase cannot initiate synthesis of polynucleotide. They can only add nucleotide to the existing 3' end. Okay, and you will see a picture. It will make sense. The initial nucleotide strand is a short RNA primer. I already talked about that. The initial nucleotide strand is a short RNA. It's called primer. Enzyme called primase can start an RNA chain from scratch and add RNA nucleotides one at a time using the parental RNA as a template. And then, as I said, this one on the bottom, I'm going to read it to you. The primer is short. This one, I already said that. This one, the primer is short, between 5 to 10 nucleotide. And 3' end serves as the starting point for the new strand of DNA. Synthesizing a new DNA strand, enzymes called DNA polymerase catalyzes elongation of new strands at a replication fork and you see a picture of it here in a minute. Okay, the most DNA polymerase requires a primer and a DNA template strand. The rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells. These numbers, do not worry about them. I should have deleted them, but if you want to know, it just gives you some idea how fast things can happen. Okay. Oh, another right now. I thought we'd get to pictures. Each nucleotide that is added to the growing DNA strand is nucleoside triphosphate. Oh, yes. Okay. DATP is similar to ATP, but it is DNA. In ATP, the ribose was oxygenated. But this one, since we are dealing with DNA, that one is, that's what the D stands for. D, it means that adenine is the ribose is deoxygenated, not the adenine. The ribose is deoxygenated. It does not have oxygen. And I told you the differences between RNA and DNA. RNA, the ribose has oxygen on carbon number 3, 4, 2, I guess number 2, yeah, if I recollect. Okay, then DNA on carbon number 2 has no oxygen, it has just hydrogen. That's what that D stands for. DATP supplies adenine to DNA and is similar to adenine ATP of energy metabolism. The difference is that sugar, DATP has deoxyribose while ATP has ribose. As each monomer, some of you had problem with this during last exam. Monomer, it means one, and polymer, it means many. Monomer, it means one. The DNA is a polymer, right? Adenine is a monomer. Guanine is a monomer. Okay, of course, the nitrogenous base, when they are attached to sugar phosphate, is a monomer. As the monomer DATP joins the DNA strand, it loses 2 phosphate groups as a molecule, and then right here. What am I saying? This one, it has 3 phosphate. DATP has 3 phosphate. 2 of them are going to be lost. 2 of them are going to be lost. And then what happens? This phosphate attached to that sugar molecule, to that deoxyribose. Am I making some sense? And of course, the sugar here is also deoxyribose. That's what that last diagram, last write-up is saying. Okay. Antiparallel elongation. The antiparallel structure of the double helix affects replication. DNA polymerases add nucleotides only to the free 3' end of growing strand. Therefore, a new strand can elongate only in 5', 3' end. Okay, so I already talked about this. 3' end is the one, you remember I said this is 3' end, and then you're adding from 5' to 3' end. Let's look at it. We'll look at it together. With all of these write-ups, if you have these write-ups, and of course your textbook, and on one side, and you have the pictures, the PowerPoint pictures of your textbook, all on one side, and read them and follow them, it will make sense. Okay, so let's go over them. And then, of course, you have the video on YouTube, and you have everything. Great, let's go. Are you guys ready? So what you have right here is the template. The light blue is the new strand. Am I making some sense? Another thing which we didn't talk about it yet, I'm going to go talk about it here in a minute. You have a leading strand in the new DNA. You have a leading strand, and then you have these fragments of DNA. You see, that's a fragment of DNA, that's a fragment of DNA, that's a fragment of DNA. And I give it the name in a minute, angle. Same on this side. You have fragments, and then leading strand. It's called, these fragments are called lagging strand. They're called lagging strand, and this is called the new strand. Okay, and as I said, we already said, the duplication of DNA is in both directions. It's happening in both directions. This is the bubble. We are talking about what is happening in the bubble. right? A little bit. So what happens here, guys? You have DNA polymerase, of course, 3 starts synthesizing leading strands. So that's what DNA polymerase 3. You have the primer, 5 prime, 3 prime is the template, DNA, that's DNA. The orange is your RNA. As I said, 5 to 10, 1, 2, 3, 4, 5. After the 6th one is DNA. Am I making some sense, everybody? So you have a short strand of RNA, and the enzyme primase makes up this one right here. Okay, then you have these binding protein molecules, so the 2 DNA do not come together. Okay, and then DNA polymerase 3, there is DNA polymerase 1, now we'll talk about that. DNA polymerase 3 is adding the new nucleotides to the, based on the template. If it is A here, it's T here. If it is G here, it is C here. Am I making some sense, everybody? If it is A here, it's T here, and so on and so forth. The law of base pairing, which I wrote it on the board on last time. And then on the bottom, what enzyme? Continuous elongation. It's continuing. This is the leading strand. Leading strand is longer, and then these are lagging. They call them the short fragments. Is called lagging strand. Right here, let's look at the animation.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The 2 strands would naturally tend to be aligned, but are held apart by molecules of a single strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction.
5'
The
to 3'.
strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
Okay. So pretty much the videos and me are going little by little. We haven't given you a complete picture yet. Okay, along one template strand of DNA, the DNA polymerase synthesizes the leading strand continuously moving toward the replication fork, leading strand. To elongate the other new strand called the lagging strand. Remember, DNA replication is one side, both sides, right? One side is leading strand, right? The other side are little fragments. Do it make sense? One side, that's one template up here, one template up here too. Same thing, leading strand here and fragments here. Stephen, are you with me? Okay, let's go. To elongate the other new strand called the lagging strand, DNA polymerase must work in a direction away from replication fork. The lagging strand is synthesized as a series of Okazaki. You will see it. Alicia, you will see Okazaki. Do you know Okazaki? Anybody know Dr. Okazaki? He was from Japan. He discovered this first. So they call him Okazaki. It rhymes, I think. It's a very nice name. If I had another daughter, I would call it Okazaki. No? Okazaki fragments, which are joined the DNA, they're joined together by DNA ligase. Okay. These lagging, do they have Okazaki? Yes. Okazaki. Lagging, these strands are called Okazaki strand. This is leading strand, no question asked. These are called Okazaki strands. And then RNA primer, origin, of course. What else? DNA polymerase. DNA polymerase makes Okazaki fragments. They're making Okazaki fragments. And then right here, what else? There's polymerase 1. You said this is, you remember DNA polymerase 3? 1, 2, 3. It makes new strands of DNA, but DNA polymerase 1 replaces RNA with DNA. You cannot have, when this is all over, this RNA must be removed. You cannot have it here. There might be a uracil here, right? Uracil. So there, who removes this and put the actual DNA in there? It's your DNA polymerase 1. DNA polymerase enzyme Stephen, DNA polymerase 3 puts new DNA, both the leading strand and the lagging strand, all of this. And DNA polymerase 1 comes in and removes these primers and put the DNA. And then finally, there is one enzyme. Did he mention it here? Yes, he mentioned it here. That is the last one. I believe this is the last enzyme. DNA ligase. Do you know what ligation means? What is the term ligation means in English language? Look, you never heard of tubal ligation? Women, they go ligate their tube. You never heard of that? You must be kidding me. Which era or which century are you from? Reno, have you heard of that? Females do tubal ligation. Females who do not want to become pregnant anymore and have babies anymore, they go to doctor, and the doctor will ligate their fallopian tube, oviduct, if you would. Stephen, you didn't know that. See, that's why you came to school today, to learn about females' tubal ligation. Later on, when you get married, you do not want to have children anymore. You tell her, of course, and she goes to the doctor. You can do the same thing with your testes, but we will not go there. That's for next lecture. That's for next lecture. Today's lecture is called tubal ligation. Ligation, it means tying up. Tying up. So what does DNA ligase will do? All of those Okazaki fragments and all of the leading strand. See, there is a space, there is a gap, right? Well, there is no gap. There is DNA in here. The Okazaki must be attached to the leading strand. Okazaki must be attached to Okazaki. Okazaki must be attached to Okazaki. Okazaki must be attached to leading strand. What enzyme is responsible for that? DNA ligase. There is no gap. Am I making some sense, everybody? You all know what I'm talking about? Okay. I think that's the last enzyme. We'll see the list. And then right here, over origin of replication, right here, lagging strand, Okazaki strand, DNA is going in both directions. Right here, primase makes up the primer, okay? And then of course, if it is 5 prime, it goes 3 prime to 5 prime in, and so on and so forth. Right here, elongating, so on. DNA polymerase 1 makes Okazaki fragments. DNA polymerase 1 replaces replaces RNA with DNA, and of course the bottom ones are not being seen. Let's watch this video.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment.
That's a leading strand.
The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments.
Oh,
Next,
okay.
each RNA primer is replaced with
Start
DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary
Polymerase
DNA
3.
nucleotide to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments.
That's DNA polymerase 3.
After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA.
DNA polymerase
Next, the enzyme
1.
DNA ligase links the Okazaki fragments to form the lagging strand.
Oh, I don't. Okay, right here. I think we talked about everything in here, and I don't see anything. DNA ligase is on the bottom. Right here, puts the fragments of DNA together, and DNA polymerase 1 removes the RNA and replace it with DNA. And again, this is helicase. Remember that unwinds DNA and unzip DNA. Here we go. Same thing again. Most of these are repetition. Another animation. What is this one about?
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles, which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
And of course, these are ligases are coming in. These are ligases are coming in and putting the DNA together. We already watched this, huh? No, we didn't watch this one. Okay, let's watch it.
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Here are the enzymes, the one that I send you, and all of the other protein molecules that are involved. He is more wordy here. The one that I sent you was quick to the point, tried to summarize it even more than this. This is summarizing it very good, but look at both of them. I don't know what happened. I thought I saved it in my PowerPoint. I don't know where that is. The one that I emailed you. Okay, the DNA replication complex, the proteins that participates in DNA is a large DNA replication machine. The DNA replication machine may be stationary during replication process. I thought I saved all of these. The real in parental extrude. The trombone model. It is kind of important that you should know. Again, DNA is a long strand right here. And the model is called trombone. What is trombone? Anybody? Alicia, do you know what is trombone? What is it?
Is it a musical instrument?
It's a musical instrument. It's a musical instrument. And look at this. It's wind down, and the protein molecule is connecting protein molecules. So it will combine helicase to your DNA polymerase. This protein molecules connecting protein molecules, everything else you study. So this loop right here, if you would, this loop right here is called a trombone model. So they're saying, so DNA is not floating all over the nucleus, not nucleus, the interface of the nucleus. This is what happening, the S phase of the nucleus. So they call this a trombone model. DNA replication.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA.
Right here, that's connecting protein molecules between polymerase 3 and the helicase. You remember what I'm talking about? This is a trombone model. She's trying to show you the trombone model. Okay?
As we zoom in, it's for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces.
This seems like a new strand, leading strand. Oh, no, here is another one. Okay, this is the Okazaki strand. That was the Okazaki.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA.
That's DNA polymerase
However,
1.
it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, proofreading and repairing DNA, there are mistakes happen. There are mistakes. If this is A, this is T, G, C, am I right? C, G, T, A, T, A, T, C. Is this correct? Is T, C correct? No, it happens. What happened, DNA polymerase, it comes and proofread. Proofread A to T, G to C, C to G, T to A, T to A, T to C. No. How can he fix it? Try. How does he fix it? DNA polymerase. Oh, there are two ways, right? Either remove this one and put G here, or, am I right? Or T stays, remove this one and put G here, put A here. Do I make sense? There are two ways to fix it. Which one is the right way? DNA polymerase does not know. Does not know. So it can fix it either way. But the question is, what was the original DNA? Was original DNA T to A, or was it G to C? Original. You know what I'm talking about? Originally it was T to A or G to C. If it was T to A, it corrected perfectly, just like original DNA. If it was not T to A, and then if it was T to A and he's fixing it as G to C, then you have what? Mutation. When you talk about mutation, right here, that's mutation. We talked about it from beginning of semester until now. Now today you get to know. So, mismatch. DNA polymerase proofreads new DNA replication in correct nucleotides. In mismatch, repair of DNA, repair enzymes correct error in base pairing. DNA can be damaged by exposure to harmful chemicals, physical agents, cigarette smoking, x-rays, alcohol. He didn't mention that. It can also undergo spontaneous changes. Here it is. You have 3', 5' nuclease. The enzyme that take off this damaged DNA is called nuclease. Nuclease is the enzyme that takes off the damaged DNA, and of course DNA polymerase comes and put the new DNA in place. And of course DNA ligase put them together. Evolutionary significance of altered DNA nucleotides. Error rate after proofreading repair is low, but not zero. Spontaneous changes may become permanent and can be passed to the next generation. These changes, mutations, are the source of genetic variations upon which natural selections occur. Replacing the ends of DNA nucleotides and limitations of DNA polymerase creates problems for linear DNA of eukaryotic chromosomes. The usual replication machinery provides no way to complete the 5' end spread, so on and so forth. It's not a problem of prokaryotic, most circular DNA. Okay, so this is reviewing what is happening as far as the new strand, old strand. Eukaryotic chromosomes, DNA molecules have special, okay. Have special nucleotides sequence at their end, right here. I'm sorry, I was moving on. This end, every time that DNA duplicates, a piece of end of the DNA erodes away, get cut off. So as you're aging, it seems like it, as you're aging, your DNA is becoming a little bit shorter and shorter and shorter. Those ends, these ends are called telomeres. Okay, telomeres is the end of the cells right here, telomeres. Okay, so it has been proposed that shortening of telomeres is connected to aging. So DNA of a 60-year-old man and DNA of a 16-years-old boy, man, is different. I want to make you some sense because of the telomeres. If that is true, Amir, what will happen to the sperm and egg? Do their DNA shorten up as they age? A 60-year-old man's sperm and a 16-years-old boy, or I should not say boy, man, young man, sperm, are they different? The answer is really no. The length of the chromosome, because you're losing genes, possibly could be losing genes. So the answer is no. And what is, here is a picture. What happened to all of the changes I made to the slides? I guess I'll have to check where my original slides are. This is a telomere. This is a chromosome, telomere, telomere. By a technique, they were able to make them lighten up at the end. Okay, so that's what chromosomes, telomeres is. What happens, the enzyme telomerase in sperm and egg, so egg of a 45-year-old woman, I'm not going to talk about the woman who went through menopause, egg of a 45-year-old woman and an egg of a 10-year-old girl, they are having the same kind of DNA. They do not undergo the erosion, cutting of the telomeres, because they have the enzymes, telomerase, does not allow this erosion to occur. There was a program on TV a long time ago. So is this the key to keep human young? If we give bottles of telomerase to people to drink, so the chromosomes will not shrink as they age. It's under intensive research right now. Maybe they found the key to not aging. Telomerase. Maybe that would be a good topic for you guys to get your PhD. Tron, do you want to get your PhD in there? Maybe. Chromosomes consist of DNA molecules packaged together in proteins and double-stranded eukaryotic cells. Supercord. Excuse me, I cannot get rid of this. But you know, let's talk about the last portion of this chapter. I talked about this. This is DNA wrapped around, it goes around, coils around.